Nutrigenomics, page number 478, chapter 27. So first, understanding the basic terminologies of what are genes, genetics. Okay, genetics means it's a study of how disease conditions or whatever you inherit from your parents, from the father and mother. What is the pattern of inheritance? When you study that, that is what we call as genetics. Okay, what is genome? Genome is the entire set of genetic material, which is DNA. Okay, and every cell every cell in your body has a nucleus. Inside the nucleus, your DNA, your genome is there. Okay, you can see here in this image here. Okay, this is a cell. Inside the cell, there is a nucleus, and inside the nucleus, you can see the entire set of genome. Okay, does anyone know how many set of genome is present in humans? Yeah, there is 23 pair of chromosomes. Okay, which means 20 or 23 pair of chromosomes, so 46. Okay, so okay, so this 46 chromosomes, they become your entire set. Okay, they become your genome. This 46 chromosomes are your genome, are the human genome. Okay, which means even if you get one cell, with that one cell, is it possible for you to determine, is it a man's cell or a woman's cell? Is it a male cell or a female cell? If a random cell is given to you under microscope, you have to check, okay, you have to do genetic analysis. So based on this fact, is it possible for you to identify if it is a male cell or a female cell? Because every cell has your genome. Yes, this XX and XY, that is the 23rd pair, and which determines the gender, which determines the sex of the organism. Okay, in humans, it is the sex of the human. Okay, and this XX and XY chromosome is also present within every cell. Even if you get a single cell, based on that, it is we can identify if it is a male's, is it from a male's body or from a female body, because every cell in your body has the copy of this 23rd pair, which is a sex chromosome. It could be XX in females, XY in males. Okay, so all these 23 pairs, okay, all these 23 pairs, the genome is present in every single cell. Every single cell has the nucleus. Every single nucleus have the genome set. Okay, is it clear to all? And what is DNA? DNA makes up the chromosome. Okay, DNAs come together and they make up your chromosome. 43 chromosome makes up 23 pair, and this 23 pair is your entire genome set. If you split the DNA, you get RNA, because double helix structure is DNA, single helix structure is RNA. Okay. So far, is it clear to all what is genome, what is chromosome, what is DNA? So the functional piece of DNA is called gene. Okay, you can see here, small genes, 2-3 genes come together and they form a DNA. DNAs come together and DNA is like a long tape. Okay, it's a very long tape. Okay, a very, very, very long tape. That is a DNA. When DNA condenses itself, it forms into chromosome. Okay, you pick out the chromosome, and if you try to unwind it, if you try to unwind the chromosome, you get the entire strand of DNA. Okay, small genes, two, three genes come together. They in different settings. Okay, and they form this entire long double helix tape called DNA. DNA is coiled up properly into the it is tightly coiled up properly, and that is what forms the chromosome. Chromosomes come together, 46 of them come together, and that's your entire genome set. Okay, so a single piece of functional piece of DNA is called genes. So genes come together, and that's when you get the entire DNA. Okay, and it is made up of few 100,000 base pairs. Base pairs of genes are different. It is too technical. You can see here, if it is possible for you to understand, see AT, TA, GC. Okay, here TA, CG, AT, AT. So there are different combinations. Okay, there are different permutations and combination of this ATCG. Okay, ATCG is the basic protein. Okay, it is a basic protein based on which the entire genes are made. Gene systems, they come in different permutations and combinations. Okay, and that's how you get the DNA. Okay, and it is non-repetitive sequence. No, there will be no repetition in this DNA sequences. The way how this ATCG, AT is placed, there is no repetition in that. Okay, and genes are located on the chromosome, and the study of genes is called genetics. Okay, all the haploid set of DNA is called genome. So all the chromosomes come together and they form the genome. There are more than 6.4 billion bases of genome. And again, these are also not repetitive. If at all it is repeating anywhere, it means you have a genetic disease condition. And where do you find the genome? It is found inside the nucleus of the cell. Okay, and the study of genomes is called genomics. So this is the basic difference between genes and genome. Okay, genes come together and they form DNA. DNA, the DNA is the long tape which condenses. Okay, and that's when you get chromosome. Chromosomes, 46 pair of them. That's the entire genome set. Okay, so genome is much larger than a gene. So you can see here, if you split the DNA into two equal halves, you get RNA. DNA is the double helix structure. Okay. RNA is a single helix structure. Okay. DNA, deoxyribonucleic acid. RNA stands for ribonucleic acid. And you can see here, the C, A, T, G, whatever was there, that is a protein. Okay. Amino acids, these are amino acids and sugar base pairs. Okay. So they form these combinations, the cytosine, guanine, adenine, and thymine. They form the different combinations, okay, and they have to form non-repetitive combinations. If at all it is repeating anywhere, it means some mutation is there, some genetic disease is there, some unhealthy changes there. Okay, so this is the meaning of DNA and RNA. Okay, RNA has to find another tape, this long tape-like structure. That's when DNA is formed. Okay, and these are the base pairs: cytosine, guanine, adenine, thymine. They are the base pairs. So protein is required for these base pairs to be built. Okay, so protein is required for your DNA to be built. Okay. So far, is it clear to all what is DNA, what is RNA, gene, genome, chromosome? Where is it found in the cell? Is it clear to all? So what are haploid and diploid cells? Okay, haploid cells are single set of chromosomes. Okay, they are 23. Diploid means 46. There are double set of chromosomes. You can see here in the previous image, here, this chromosome, what do you think? Is it a haploid or a diploid? This chromosome which I am circling with the mouse, is it a haploid structure or a diploid structure? It is a diploid structure because it has the pair. Okay. You have 23 of this pair in your nucleus. Okay, this is a diploid structure. When you split it in half, that's when you get a haploid structure. Okay. So what happens in Down syndrome? You must have heard about Down syndrome. Okay. There is another name for Down syndrome, which is called trisomy 21. Trisomy 21. I have put it in the chat box. Trisomy 21 means Down syndrome. Okay. So what is this trisomy 21? See, this should be, see, 46 should be there, 23 pair, 46 should be there. In trisomy, what happens is that the 21st chromosome, okay, the 21st pair of chromosome, instead of having these two structures, instead of it being diploid, it has one additional structure as well. So it has three, okay? So 21st chromosome has three chromosomes, okay? It has three somes, it has three cells, okay? So it becomes trisomy 21. That is your Down syndrome, okay? Any extra pair into this chromosome, into this diploid chromosome, that will create a genetic condition. Just an example, so that you understand what is normal and what is abnormal, okay? So coming to the terminology of nutrigenetics and nutrigenomics, okay? Nutrigenetics, we'll see nutrigenetics first. Your genes affect how your body responds to food. Your genes affects how your body responds to food, okay? So there are 57 participants in our class right now, okay? If I give peanuts to all of you, okay, understanding that everyone can eat peanut, okay, but is it true? Will everyone respond to peanut in the same way? Everyone's body will respond to peanut in the same way? No, right? Why? Because each one of you, your genes, the genes that you have received from your family, that affects, okay, that affects how your body will respond to peanuts. That is nutrigenetics, how your body is responding to food. Okay. Coming to nutrigenomics, because it is individualized. Everyone's genes does not act in the same way to different food. People are lactose intolerant, okay? Some are lactose intolerant, some are allergic to peanuts, okay? Some are foods sensitive to specific kind of food. Not everyone can have seafood. Why? Because your genetic makeup is in such a way that your body may show some malfunctioning when you expose it to certain kind of food. Okay, that is nutrigenetics. When you study about this, how different people's body is responding to food. Okay, so that is what we consider under nutrigenetics. Coming to nutrigenomics, next terminology, what you eat affects how your genes behave. Okay, what does it mean? For example, if on a daily basis, you are having some processed food, every one of you, 57 of you, every one of you are having the same processed packaged food on a daily basis, will it cause some genetic change in your body after certain time? after some time. Some or the other way, it will affect your genes. Some or the other way, food can change your genetic makeup. Now you understand the difference between these two words. How your body responds to food. Genetic makeup is already there. A genetic makeup is already there. You are giving your body some food. Your body is responding in a certain way. That is nutrigenetics. Okay. But there is another story. Some specific food can change your genetic makeup. Some food metabolites, some preservatives, okay, uh, some pesticides in the food, they can change your genetic makeup. Okay, food can also change genetic makeup. Food can cause genetic mutation. And when you study that, how this food is leading to this cancer, how this kind of food is increasing the risk of genetic conditions, that is nutrigenomics. Because you have to study the entire genome. Okay, the entire genome of that individual or the community has to be studied. How this entire genome is responding to this food or the metabolites or any substance in that food. Okay, addictions as well, substance abuses as well, like cigarette smoking, drug abuse, alcoholism. Okay, these can also cause some genetic change. Okay, this can also make your genes behave in a certain manner. Food additives as well, preservatives, additives, coloring agents, some pesticides, fertilizers used in agriculture. Okay, so is it clear to all the difference between nutrigenetics and nutrigenomics? Environmental pollution as well. Okay, but environmental pollution does not come under nutrition part. Okay, that is the pollution part of it. That is external epigenetics. We call it as epigenetics. Okay, how your entire environment where you live in. Okay, how does it bring the change in your body? That is epigenetics. Okay, that is a different. We will come across that. It's a different story. So is it clear to all? Yeah, these genetic changes, which is happening right now, like food-induced genetic changes that is happening right now in your body. Tomorrow, if you are expanding your generation, these genetic changes can also be inherited by the next generation. So coming to the principles of nutrigenomics, okay? What is nutrigenomics? You are studying how nutrition affects your genetic makeup. How nutrition affects your genetic makeup. Okay. So gene expression can occur directly or indirectly. Okay. Like gene expression means some change in your genetics. Okay. Some mutation, some change in your genome can occur directly or indirectly, which means some preservatives that you eat on a regular basis, it can directly cause a genetic mutation in your genome, or it can cause some inflammatory reaction. Okay? And when you are having this inflammatory reaction on a prolonged basis, okay, this inflammation can cause some genetic mutation. For example, colon cancer. Okay. Are you aware of the association of red meat and colon cancer? years after years, studies have proven that overindulgence with red meat, okay, like beef, mutton, okay, they come under pork, okay, they all come under red meat category. Lean meat is the white meat, poultry meat, chicken, duck, okay, they come under the lean meat, the poultry meat, white meat, okay. Red meat is beef, mutton, pork, okay. So, in communities and even in Western countries where red meat is a staple diet, okay, we have seen a lot of colon cancer predominantly rising in these countries, okay, wherever the population regularly eats red meat, prefers red meat over white meat, okay. So, the red meat may not be causing the this colon cancer gene mutation directly. It may be triggering the intestine in certain way. These triggers are causing the gene mutation. Okay, so this is gene expression can happen directly or indirectly. Some chemicals can directly go and attack your DNA. Some chemicals will not directly attack the DNA. It will cause some signs and symptoms in your body. And when these signs and symptoms are happening in your body on a very regular basis, okay, that can cause some gene mutation. Okay, that is the first principle. Then second is diet. Diet is a risk factor for certain diseases in some individual under certain circumstances. The same red meat, association of red meat and colon cancer. Okay, and having high fat diet, having high saturated fat in your diet, definitely will put you at the risk of cardiovascular diseases. Okay, so definitely diet is a risk factor for certain diseases. High saturated fat diet, high protein diet will have the risk of kidney-based disorders, nephrological disorders. Okay, so diet always has a risk factor. No, with vegetarianism, colon cancer risk reduces. Okay, the risk of colon cancer is very low in vegetarians as compared to non-vegetarians. Okay, there could be other chemicals in the food, like the way how you are procuring your vegetables, fruits, okay, some preservatives, additives, fertilizers, pesticides used in agriculture that could lead to some other disease conditions. But if you compare non-vegetarians and vegetarians, the risk of colon cancer is always higher in non-vegetarians as compared to vegetarians. Then chronic disease onset, the incidence, progression, or severity are influenced directly by your diet regulated genes. Okay, for example, there are certain genes in your body that control your metabolism. Okay, and if these genes are affected, okay, these genes which are supposed to control the metabolism, okay, if these genes are affected, automatically you will face the situation where a chronic disease has begun in your life and it will last long. And there usually will not be any cure to that rather than restricting yourself from that diet. So that is the association of chronic disease onset and diet. Certain diet can specifically affect the genes that are responsible for your metabolism. And if these metabolic genes are getting affected, your chronic disease will start. Then your genetic makeup. The influence of diet on health and disease is likely to be affected by genetic makeup. For example, if you take a if I give you two diabetic patients, one is a South Indian, one is a North Indian. Okay. So, will you give the same diabetic diet to both of these patients? No, you will not, because you may say rice is bad for diabetes, but rice is more bad for the North Indian who is diabetic than the South Indian. Okay. Millets are common for everyone. Ragi, jowar, bajra. Okay, it's common for everyone. Millets are good for all the diabetic types. But wheat is also equally responsible. If the South Indian diabetic patient, if he leaves rice and start eating wheat, do you think his diabetics will be under control, or it will get more aggravated? It will get more aggravated. Okay, because the genetic makeup is is presumed to digest rice more efficiently than wheat. Wheat also has high glycemic index, but we have a myth, there is a myth running that if you start eating chapatis, specifically for diabetic people, if you start eating chapatis, your glycemic index will be low. It's not like that. Okay. It's more based on your genetic makeup as well. South Indians do better with rice-based recipes than North Indians. Okay, if North Indians start eating rice the way how South Indians eat, they will definitely have high glycemic index. Okay, high blood glucose level. Okay, because the genetic makeup is used to digesting wheat-based recipe, generations over generation. Okay, rice and wheat only have very slight variation. Okay, there is no big variation, but there is a myth going on that wheat is better than rice when it comes to blood glucose control. It's not true. If even rice has variance, if you go and eat red rice or brown rice, parboiled rice, they are much better than wheat. Okay, they can do much better. They are much easier on your stomach or intestine than wheat. Okay. So that is your genetic makeup. This is genetic makeup, regional genetic makeup that is affecting the way how your body is supposed to digest food. Okay. Rice, if you go for brown rice, it has high fiber content as well. Okay. Refined wheat will not, but if it is unrefined wheat, has high fiber. Then nutritional intervention. This is the management part. So nutrigenomics, what you are doing is, once you understand the concept of how food is affecting your genome, how food is affecting your genes, now you can manage the condition nutritionally. Okay. Tailored to every subject. Every individual will have a separate nutritional background, separate way in which their genes are or genes are responding to certain kind of food. So how to add more food into their diet, how to remove certain food from their diet, it is customized. It is tailored to individual subject. Okay. So to make nutritional intervention also, Nutrigenomics will help you out. Okay, you have to understand what is your client's nutritional status. Are they overnourished, undernourished, malnourished? What is their genotype? Okay, what is their current health status? What is the family history of diseases which is prevalent in their family? What diseases their parents had? What diseases their grandparents had? Because when you are doing genetic counseling, you have to get all this information as well. Okay. So these are the 5 major principles of nutrigenomics. Next, coming to interactions. Interactions in detail they have given, but you don't require that in detail. You just have to just understand the direct interaction, epigenetic interaction, and genetic variation. That's it. So direct interaction, you can see here in this image. So there is an activator, okay? Activator could be any chemical, okay, any chemical, any enzyme. And every DNA strand has a site where this activator can go and attach itself. The chemical or this enzyme can go, or it could be a protein as well. These activators can be a protein, a chemical, a metabolite, an enzyme, anything. Okay. So there are specific portions in your DNA, parts in your DNA, where this activator can go and attach itself. Okay. And when this attachment is successful, okay, that's when new RNA, new DNA, okay, or any target gene will get the message and some change will start. Okay, so this is a way in which nutrition, some vitamins, some minerals, okay, some nutrients can also become activators. They can directly go and attach to your DNA. And once this attachment is successful, okay, a new transcription will start. Transcription means a change in the gene. Okay, a change in the gene will start. Okay, once the activator, sometimes this activator is nutrients as well. They go and attach to the DNA, and once the attachment is successful, in the entire DNA strand, some changes will start. Okay, there will be a gene that could be a target, and that target gene will undergo some mutation. Okay, and this is how your body prevents this activation, prevents this transcription, because there are repressor genes within your body. Okay, these repressor genes are part of your immunity. Okay, what is their job? Their job is to go and bind themselves on the binding site of repressor. There are binding site for activator, and there are binding site for repressor. Okay, so this could be same site, this could be different site, because every DNA genes are different. They act in a very different way. Okay, some in some strands of the DNA, the binding site for the activator and repressor is the same thing. Sometimes the repressor will come first and bind itself on the DNA. There is no space for the activator to come and bind. There will be no change in the DNA. You will have a healthy DNA. No mutation will take place. Okay. And in some DNA, even if the activator has come and bound itself, even if the activator has come and attached itself to the DNA, there will be some other site for the repressor to go and attach itself, completely different parts of the DNA. But once the repressor have also attached itself, how much ever activator comes and attaches, there will be no change in the DNA. Repressor is trying to protect the DNA from any mutations. Okay, it is trying to protect your genes from any mutations. You can see here, the repressor has successfully attached itself to the DNA, and there is no transcription taking place. There is no change taking place. So the target gene is as it is. No change has occurred here. The target gene is as it is. Okay, so this is the direct interaction. Okay, this activator can be certain nutrients, certain chemicals, certain fertilizers, pesticides that has come into your system. They can go and attach to the DNA and cause some change, serious shift in your genetic makeup. Okay, but if you have the repressor genes working, if your immunity is strong, if you have your repressor genes working, they will attach themselves to their specific site in the DNA, and they will not allow activators to attach anywhere, and no change will take place, and your DNA will be, your genes will be healthy without any mutations. Okay, so this is the direct interaction. Is it clear to all? Then epigenetics. Epigenetics, as I mentioned, this is the environment where we live in. Okay. The most simplest example is people who live in the cities, urban area, and people who live in rural areas. They have completely different epigenetics. Okay, people who live in urban area, they have more factors that can cause genetic mutation in them. Environmental pollution, noise pollution, air pollution, water pollution, lifestyle, access to unhealthy junk food, lack of physical activity. Okay, so these are the epigenetics, how the environment, things in the environment, external environment where you live in, the society where you live in, how it affects your genes. Okay, so you can see here the link between the different lifestyle in an urban area, lack of physical activity, pollution, okay, smartphones, televisions, junk food, okay, it's causing some genetic changes, and because of this genetic changes, heart diseases are high in urban population, diabetes is high in urban population, cancer, rate of cancer is high in urban population, metabolic syndromes, rare diseases, these all are high in urban population. And also the cost of healthcare is also shooting up in urban population. So this is your epigenetics. Okay, is it clear to all? How epigenetics, metabolism, and your genetics, and how you respond, how you get different disease conditions are related. Then we have genetic variation. So genetic variations, air pollution can be a direct interaction. Depends on what exactly is the pollutant in your air. Okay. Air pollution is everywhere, even in rural India, you have air pollution. But what is the content of the air pollution? Okay. Is it industrial? Is it just dust? Is it smog? Is it fog? Okay. That is important. That to understand that concentration is important. Okay. COVID-19, we can't be sure of because still researches are going on in COVID-19 variant. Okay, how is it? And all the patients who have been affected with COVID, okay, how is it once you got contracted with COVID, and next what happens in your next 10 years, 50 years, what's going to happen? We don't know yet. Okay, some interaction must have took place among the people who had caught COVID. Genetic variation, it's normal as you age. Okay, some genetic variation can take place, okay, which are completely out of your control. Aging is one of the causes of genetic variation. Change in lifestyle, stress, okay, with age your stress level increases, okay, that could also cause some genetic variation. So these are common genetic variation which can happen to anyone. Everyone has to go through the process of this life, and in these different various processes of life, some genetic variation can take place. Some could be harmless genetic variation, others could be harmful genetic variation. Next, coming to the tools used in nutrigenomics. So understand what is genotype and phenotype. Okay, genotype is like, let's say, XX and XY. Okay, that is genotype. It's in code. It's in code words. XX, XY, it's in code words. What becomes the phenotype? XX is a female, XY is a male. Okay, so genotypes are your gene expressions. Phenotype is the physical features which you can actually see. The genotype of eye color, okay, different eye colors are completely different codes. But the phenotype you will not say it in code. You will say blue colored eyes, black colored eyes, gray eyes, green eyes, hazel eyes. Okay. So these becomes the phenotype. Physical characteristics becomes the phenotype. But the code behind it, that is the genotype. Okay. Every person who has blue eyes, okay, everyone in the world, whoever has blue eyes, they all share the same genotype of eyes. Everyone who has hazel eyes, they all share the same genotype of has hazel colored eyes. Okay. Everyone who has blonde hair, they have a specific genotype. Everyone, those who have brown hair, they have another genotype. Red-haired people, they have another genotype. Okay. But the phenotype, the physical way of how it comes up is the same. Okay. So the terminology is used in the tools of nutrigenomics. So you have genomics, which we have seen earlier. It's the study of the entire genome. Genome means when you are studying the 46 chromosome, that is 23 pairs, and 46 chromosomes when you are studying about it, okay, that is called genomics. There are more than 25,000 genes, still counting. Now it is more than 30,000, okay. So how the information is stored in the genome, okay, how to predict what can happen, how to predict the next generation with, if there is a husband and wife, with their genome, you can predict what kind of genetic disorders they can pass on to their next generation, okay. So studying all this is called genomics. Next is transcriptomics. Transcriptomics is, as I mentioned earlier, transcription means a change. Okay. DNA, when it gets activated because of some activator, okay, transcription starts. Okay, RNA will start this transcription. RNA is the single strand. DNA is the double strand. Okay, to start a change, even a single strand of RNA is more than enough. The RNA can start the transcription because with its base pairs, single base pairs, just the base proteins, whatever it has, not even in pairs, just the bases, whatever RNA has. With that, a transcription can be started. So when you study about these changes, how RNA, how transcription takes place, okay, studying about the RNA in detail, studying about the bases in RNA in detail, okay, the sequences, different sequences of the bases in RNA, when you study about all these things, it comes under transcriptomics, okay. That is another tool. Then proteomics. When you study about the protein molecules, if you see in the first slide, if you see cytosine, guanine, adenine, these are the molecules, the amino acids and sugar bases, okay. They are the ones who are making this base pair. When you study about these base pair, cytosine, guanine, adenine, uracil, thymine, okay, when you study about these base pair, that is also called, that also comes under proteomics, okay. And there are more than 1 million protein structure present in the DNA, which studies are still going on under them. Then we have metabolomics. So study of the molecules involved in cellular metabolism, and that would eventually depict the phenotype of an organism. That is called metabolomics. Okay. Even nutrients that undergo metabolism, and after metabolism, whatever remains, which we call metabolites. Okay, metabolites are after metabolism, the byproducts, whatever remain. Okay, the smallest byproducts, whatever remain. These metabolites can also go and cause some changes in the DNA. Okay, harmful or even good, some changes in the DNA can take place because of the metabolites of metabolites of the nutrients. Okay, so studying that also becomes another tool which is called metabolomics. So these are the different tools used in genomics, transcriptomics, proteomics, metabolomics. So these are the different tools used in nutrigenomics. Is it clear to all? In detail, genomics, proteomics, metabolomics is given in the textbook, but that is not required from exam point of view. Just understand the single line meaning of it. That's it. Okay. Next topic is bioactive components of food. Page number 484 onwards. So bioactive components means these are specific, for example, antioxidants. Okay. Antioxidants is the best example for bioactive component. Okay. Functional component in food. So what they do, they are in some or the other way helping your body to stay away from genetic mutation, to keep your DNA, your gene, your genetics, your genome healthy. Antioxidants is one example of it. Okay, proteins, antioxidants, lipids. Okay, they all are bioactive components found in your food, which is helping at your cells at cellular level. It is helping your DNA to stay healthy, to stay away from mutation, to stay away from tumorous growth, cancerous growth. Okay, it represses cancer growth. How? We'll see. So one way is the bioactive components in the food. It can control the cell growth in a normal way. Okay, which means when cells are growing and differentiating in a normal pace, you have a healthy body. But when cells are growing and differentiating in an uncontrolled way, in an abnormal, uncontrolled way, what will you get in that situation? Yeah, antioxidants also act as a repressor in certain genes, not in all genes, certain parts of the genes. Yes, you get cancer or tumor. When cell growth is uncontrolled, you get cancer or tumor. Antioxidants and other bioactive components in the food, they can help you overcome this. Okay. Apoptosis. Apoptosis means cell death. Okay. Your cells can identify which cell is infectious or which cell has the potential to become cancerous. So what it does on its own, it will kill itself. It identifies that, okay, I am becoming cancerous. I can turn into a tumorous cell. It's better I kill myself. It's kind of that kind of technique that happens here. Apoptosis, in which the cell dies off. Okay, the cell kills itself. Okay, that process is called apoptosis. It is required. Okay, it is required so that healthy cell emerge in its place. Old cells, unhealthy cells, okay, potentially cancerous cells, they die off within your body through apoptosis. Okay. Then repairing the DNA strands, which damaged DNA strands, because free radicals are available in your body. They're always there in your body. So these free radicals can damage your DNA strands. Okay. So the repair work of these DNA strands can also be done with the bioactive components like antioxidants in your food. Apoptosis means cell death. Will kill itself once it knows that either it is infectious or it is turning cancerous or it has become worn off, old. Okay. Then inflammatory response. There are food there are certain food that are called anti-inflammatory. For example, omega-3 fatty acid rich food. Okay, those are called anti-inflammatory. Yes, skin cells are also an example of it. Old skin cells will die off through using through apoptosis. New cells will come in its place. Okay. So inflammatory response, to calm down the inflammatory response, okay, that is also important, because if you imagine one part of your body, if it is undergoing inflammation over and over again, one segment of your body, it is undergoing inflammation over and over again, that area can turn that inflammatory response, overdose of that inflammatory response can lead to cancer, can lead to some mutation, it can lead to some other disease condition. Okay, so the bioactive components in the food, if it has anti-inflammatory function, it can calm down the inflammation and it prevents the potential risk of mutation. Then hormone regulation, that is another function of food. Cell differentiation, that is old cells, okay, they multiply, okay, and they grow more in number because the body keeps growing. The specifically in young children, body keeps growing. So the cell differentiation is taking place. Even in adults, cell differentiation is taking place. Old cells are being replaced by new cells, okay, because of cell differentiation and proliferation. If when it is in a controlled way, okay, when cell differentiation and proliferation is in controlled way, you have a healthy body. When it is in uncontrolled way, you get carcinogenesis. You can see here the next is carcinogenesis. So this you see here the blue part in this part of the picture. This is impaired cell differentiation and proliferation. Okay, this is not normal. Abnormal cell differentiation and proliferation is the blue part. The other two sides you can see, majority of the cells are identical. They look similar to each other. They are growing in the same line. They are almost in you can see the different columns here, okay, on both the sides. This is normal growth. But in center, this blue part, you can see it is completely abnormal. Each cell is completely different from each other. They don't at all look like their other cells who are in the same line. So this is carcinogenesis. This is cancer. Okay, when cell differentiation and proliferation is out of hand, this is what you get, cancer. So bioactive components of food, they suppress this carcinogenesis as well. Anti-cancer carcinogenic effect certain food has, like turmeric, garlic. Okay, they have, to a degree, they have anti-carcinogenic effect. So they are also bioactive component of food. Is it clear to all so far? Examples are turmeric, curcumin, which is present in turmeric, polyphenols present in tea, green tea, antioxidants present in red grapes, beta carotene. They all are bioactive components of food. Basically, all your antioxidants, all the anti-inflammatory agents, omega-3 fatty acids, amino acids, essential amino acids, Next is chronic disease and your genetics. Okay, how is it linked? How is chronic diseases linked to the genetics? Okay, so we can start from diet first. Okay, diet, if it is unhealthy diet, okay, over a period of time, again and again, if you still indulge in unhealthy diet, it will create some changes in your gene expression. Okay, some preservatives, some chemicals, some metabolites, saturated fats. Okay, it can create change in your gene expression. Okay, and abnormal gene expression will be the result of it. Some gene mutation has taken place. And when the gene mutation has taken place, cells will be dysfunctional. Okay, every cell has its function, every organ, every tissue has its function. But wherever mutation has taken place, wherever gene mutation has taken place, that cell, that tissue, that part of the organ will be dysfunctional. And when you have a dysfunctional part in your body, you suffer from a disease. Okay. Sometimes what happens, disease can lead to all these changes. For example, you are suffering from an inflammatory disease from months and months together. Either you're not getting treated or you're not getting the right treatment. Okay. So this diseases, you have caught up with a disease. Sometimes it could be an infection. Okay. It was a long-term infection. And because of that infection, some parts of your cell, for example, TB, pulmonary TB, okay, it's a disease condition. And the patient who suffer from TB, do you think their lungs, pulmonary TB, do you think their lungs will ever be back to normal again? Those who are from the medical background, they would know. Any patient who is suffering from TB, pulmonary TB, even if it was for 1 month or 6 months, do you think the lungs will ever go back to the previous way how it was before TB? It will not be, because scarring of the lung tissue has taken place. When you suffer from TB, it scars, it forms scar in your lungs because of your calcification, mucus production, calcification. Okay. And because of that area, the lung scarring, scars will never go back to normal. Okay. Scars can never go back to normal. Just an example. So there was a disease, let's say it was TB. And because of this TB, the cells of the lungs have become dysfunctional. Certain areas of the lungs have become dysfunctional. Okay. And it has caused abnormal gene expression. Because of the dysfunctional cells, some gene mutation has taken place in your body. Okay. And because of it, your diet will be changed. Okay, you will have to have more protein in your diet, because you have to help your body recover from the scar. Okay, when the scar formation is there, your diet will change. Okay, because of TB, now you have to add more vitamin C in your diet, you have to add more protein in your diet, because you have lost a lot of weight. In TB this happens, a lot of lean mass, muscle mass is lost from the patients. You have to add more protein in your diet, more vitamin C in your diet. So this is how diseases affect your diet, and how diet affects the or leads to a disease. Okay. Is the pathophysiology clear here? Is the flow chart clear here to everyone? The sides of this image, you don't have to study about it much. You just have to understand the flow chart in between, the arrow, the green arrow which is given in between. Just understand that. That's enough. Another example we can see here. This group of people on left, they have the low genetic risk of cardiovascular diseases. Okay, coronary artery disease. in their family, they didn't had the history of coronary artery disease. Okay. This group of people you see on the right, they have high risk of coronary artery disease, maybe through family history. Okay. Both the parents, grandparents suffering from, they had suffered from coronary artery disease. So these individuals also naturally have the high risk of coronary artery diseases. Okay. So people who have low genetic risk, if they have a healthy lifestyle, their arteries will also be healthy. But even with low genetic risk, you still indulge in poor lifestyle, unhealthy lifestyle, sedentary lifestyle, okay, unhealthy eating practices, okay, unhealthy sleeping practices, okay. So your risk for coronary artery disease will increase. You will be at the risk of coronary artery disease, even though you do not have a family history of it, you do not have the genetic risk of it, but still your lifestyle can still lead you to coronary artery disease. Okay. Coming to the people who have high genetic risk, even if they have healthy lifestyle, there are certain elements of plague that will take place. Okay. It may not be harmful. Even they have to work a lot. They have to work a lot on their health because they are aware of it, that they have a genetic risk. So they take that conscious effort towards healthy lifestyle, but still the risk will linger on. There will be a certain degree of cholesterol they suffer from after certain age, after 50s or 60s. Even if they have a healthy lifestyle, the genetic risk will show up in some or the other way. Okay. It may not be harmful. But again, people who have high risk, and if they still have poor lifestyle, okay, they will definitely suffer from coronary artery disease. It will come sooner. Till grandparents, that is more than enough. Okay. Usually only after grandparents we can retain the information. Okay. Great grandparents, not, it is not possible for everyone to know the detailed history about the great grandparents. Till grandparents, if you know, that is more than enough. And grandparents from both the sides, maternal and paternal grandparents, that is more than enough. For females, you have to give the family history of other female relatives as well, like aunts, great aunts, mother's cousins, mother's sisters. Okay. For reproductive health, for females, for women, you have to consider the family history of what disease conditions other women are suffering in your family.